Good morning, children. In the previous module, we have discussed about the germination of seeds. And we have discussed about the factors that affect the germination of seeds. So, there were three factors that we have already learned. Those three factors are air, that is oxygen, temperature, that is sunlight, and water. In the previous module, we have already seen two demonstrations that explain the importance of water and temperature for the germination of seed. But today's module will discuss about the requirement of oxygen, the importance of oxygen for the germination of seed. So, this experiment has been performed with some soaked gram seed taken into two conical flasks, A and B respectively. In this slide, you can see that there are two conical flasks, A and another one is B. In conical flask A, you may see that the mouth of the conical flask is tightly stopped with a stopper and it contains plain water. And soaked gram seeds are present in both the flasks. In flask B also, we have closed its mouth and it contains water, a test tube containing water. But the water is not the plain water. In that, we have given or we have mixed alkaline pyrogallic acid. So this flask B contains a test tube that contains alkaline pyrogallic acid, and this pyrogallic acid basically has the ability to absorb the oxygen from the atmosphere. So, the air which is inside conical flask A contains oxygen, and the air present there inside the conical flask B, it doesn't contain the oxygen. Oxygen is actually absorbed by the pyrogallic acid. So, therefore, you may see that in flask A, you will see that the seeds, the gram seeds, germinated. They have germinated, or the germination can be observed in the conical flask A. But at the same time, if you see the gram seed present in conical flask B, there is no germination that has taken place. So therefore, this describes or the inference that can be made that the germination in conical flask A is proper and is positive because the air which is present there inside the conical flask contains the oxygen, but the air which is present inside the conical flask B, it doesn't contain any oxygen, because the alkaline pyrogallic acid has absorbed the oxygen from the air, and hence the seeds have not been germinated. So, next experiment deals with all the three factors, or the demonstration has been done to show that all the three factors together at the same time is responsible for the germination of seed. So in this experiment, we have taken a beaker into which we have dipped a glass slide. Okay, in that glass slide we have attached or we have tied 3 bean seeds at a fixed distance. Okay, so there is bean seed A, B and C, and there is a the beaker is half filled with water. Seed A did not germinate. Okay, and you may see that the position of seed A is completely out of water. In seed B, you may see the germination has taken place, and you may also observe that the seed B is partially inside the water and partially is exposed into the air. At the same time, if you see seed C, you won't see any germination again because it is completely dipped into the water. So therefore, the inference from this experiment that could be made is that in case of seed A, the seed is only obtaining light and air but not water. So that means out of 3 factors, 1 factor is missing, and hence the seed A has not germinated. In case of seed B, you may see that the germination has taken place properly. You may see the radicle coming out and going down towards the water, while the plumule is getting changed into a seedling and is going in upward direction. So this hence proves that seed B is getting the exposure of all 3 factors, light, air and water, and hence the seed is germinating out. In case of seed C, at the same time you see that it has not germinated again, because seed C is completely dipped inside the water and is getting only 1 factor, that is water, and it may has obtained certain diffused or certain gases which is already present there, dissolved in the water, but it is those respiratory gases are not enough for the germination of seed, and it is also not getting the proper temperature. So therefore the germination in seed C is not apparent or has not been taken place. So by these 4 experiments that we have discussed in today's module and in the previous, hence prove that 3 factors, air, water and sunlight or temperature, are necessary for the germination of seed once the seed completes its period of dormancy. In the next class, we will discuss about the types of germination. So students, your task will be you have to draw the figure number 6.3 and figure number 6.4, and you have to write down the experiment observation which is there given in the slide, and you have to write down the function of pyrogallic acid. Thank you.